Now the genome of the virus is the Flavivirus genome. The genome of the virus we already studied. It has an RNA genome with the positive sense of the virus and which are non-segmented, as you see enveloped viruses. However, now the present research, you can say there are two groups. One of maybe enveloped and one of them are not enveloped. However, from the diagram just in front of you, there is the envelope on the both sides, and then inside you can see how the capsid has been arranged and the RNA has been surrounded inside the capsid. Similarly, the RNA, the genome, is just apparent. RNA encoding to the polyproteins, and then the polyproteins on the presence of the proteases, it will be processed into the protein. There are two kinds of the protein, the structural proteins and the non-structural proteins. As in all the viruses, the non-structural proteins are called as NS proteins. So there are the different kind of the proteins. The C, PRM and E are the structural proteins, while the NS1, NS2, NS3, NS4 and then QB, 2K and the NS5, these are the, as you apparent in front of you, are the non-structural proteins. However, there are some unknown proteases are there, and these proteases are very important for the replication and for the protein to be processed. Two-group hypothesis was always there. However, these days with the structures which are obtained from the electron microscopy and from the basis of the morphological information, we are clear now that actually there are two extinctly groups are present for the flaviviruses are there. The characteristics of the both of the groups are not the same. Supported the hypothesis of the existence of at least two different groups are there. We can see two different groups of their characteristics and their properties one by one. One group include the non-enveloped viruses. The family Reoviridae, which has the genera of orbivirus, coltivirus and pseudonaviruses are included in this group of the flaviviruses. About the size of these group is about 60 to 80 nanometer, and the viruses have the icosahedral symmetry. Several concentric capsid layers are surrounding the segmented double layer RNA in this kind of the flaviviruses, while the other group includes the enveloped viruses inactivated by the ether and deoxycholate. It is about 50 to 60 nanometer in diameter. Infectious single-stranded RNA genome of the positive polarity is there. Two antigenically distinct subgroups are there, while group A and B. So the second group will again be divided and distinctly will be divided into two different groups, which we will group them as group A and group B. Now the medical importance of the flaviviruses. Mostly the serologically related to each other. It is very important property of the flaviviruses that serological point of view, they are related to each other. So we can use the antibodies from one of the virus to treat or further against the other virus. Antibodies are used for the treatment at the moment when disease is already prevalent. The vaccines are often used before the disease happens, and the antibodies or the immunogenics are used after the disease has been prevalent. So in this way, it is very important that we can use the antibodies for one virus against the other ones. Antigenic variability of hepatitis C proteins are very important. That hepatitis C proteins are very variable, and it makes them very difficult to for the immune system of the host. Immune system of the host to recognize them because they are very variable in the hepatitis C. The 17D vaccine is one of the most efficient vaccine ever developed, and it was developed against the flavivirus. So it is very nice vaccines is present, and it is important that that disease was derived against the flavivirus, and the virus was isolated from a man who recovered the infection by the virus, and the vaccine was developed.